bit more about that. At Stanford, I'm in John Pringle's lab, and we work with a sea anemone species called Aptasia, and that's shown here. And we're trying to promote this as a model system for studying coral biology and coral symbiosis. And it is a much easier animal to work with in that it does not have a calcareous skeleton. It's not as large as corals. You don't need a permit to collect them. And what's especially useful is they are easily rendered abosymbiotic. So shown here on the left-hand side is the symbiotic anemone. It has the dinoflagellates, the brown pigmentation in the host here. And on the right-hand side, after inducing bleaching and heat stress, it can remain abosymbiotic indefinitely if kept in the dark and continue to be fed and cared for. So it's a really easy system to manipulate, and that's what makes it a good model system in the lab. And what we have been able to do successfully in the lab too is to get their larvae, is to get spawning in the laboratory. And so Aptasia is a common aquarium pest actually, in that it grows like a weed in a lot of aquarium tanks because it pedal lacerates like crazy. And so that's asexual reproduction. What we have been able to accomplish in the lab now is sexual reproduction. So that is spawning, and that is producing eggs and sperm and getting larvae and doing developmental studies with them and essentially building a lot of these tools that we need for the system. And so here is a cluster of eggs at the base of the stalk of a female. And these eggs are actually abosymbiotic when released, meaning that they don't have the symbionts. And so what you see here, these symbionts are exterior to the eggs. And so they get expelled at the same time when the eggs do from the female. It just naturally does that. And here is a picture of the eggs. And so they measure about 100 microns across. As you can see, no pigmentation inside. And so they don't have the symbionts to begin with. And they don't get the symbionts until the larval stage. So that's the earliest stage that they can take up symbionts. This is the sperm here, and we have set up a clonal male line that we pair up with a lot of different female clonal lines that we have in the lab too to produce viable larvae. And I want to show you very quickly here what a larva looks like. So that's one right there, swimming across a biofilm. So this dirty mess here is a biofilm. And you can see that they're clear and translucent, so this one does not have symbionts at this time. Here's another one entering the screen now. They are much smaller than the coral larvae I'm used to looking at, but they're a lot of fun, and they can take up symbionts pretty easily, specific ones too. And so specificity is a question that we're really interested in. And so I have two main projects in the lab, and the first one is recapitulating the sexual cycle of Aptasia in the laboratory. And this is a very important tool development component, and this would allow analysis of Symbiodinium uptake specificity by the larvae. So Symbiodinium is the genus name describing the dinoflagellates, the brown symbionts that live within the host here. And with the achievement of a sexual cycle, being able to reproduce that in the lab, we'd also be able to develop some classical and molecular genetic analyses for Aptasia as well. And also, this might open some doors to methods of germline transformation with gene disruption constructs too. So it's really important to be able to get larval settlement, and it's currently a roadblock within the lab, in that we are able to get lots and lots of spawning, lots and lots of larvae to work with on a regular basis, but settlement is still an issue, in that over time these larvae end up shrinking because they're using up their nutritional resources, and then they just die. They would rather die than settle. So it's still a challenge, and it's still something that we are currently looking at to find. The progress so far is we do have regular spawning, which is a really good thing, and this has been influenced by particular light cues and temperature cues that we give to the adults to induce spawning in the lab. And the larvae can be maintained for weeks. Up to 6 weeks has been the record so far for me, and we can observe efficient Symbiodinium uptake. And right now, as I've mentioned, the big challenge is identifying the natural or artificial cues that induce larvae to settle and metamorphose. And so this is, for me, bridging what I've learned in my graduate career and try to apply it to the system and see what could potentially work for the species. And this has actually led to a lot of field work, because in the lab so far, I haven't been able to find a cue that works. And we thought a good idea would actually be to go back to the original home, where adult Aptasia are abundant, and there we can maybe find the natural cue that's responsible for settlement and metamorphosis. And here is a group of Aptasia adults growing on a biofilmed rock, and here they're growing on crustose coral and algae. And so these are both in a saltwater pond at the Whitney Marine Lab in St. Augustine, Florida, where I did a lot of these studies. I also went down to the Keys and looked at a lot of Aptasia in mangrove forests. And there are huge populations of them. And a lot of them grow in clusters like this in one location at Summerlin Key, Florida. They grow in clusters like this with other types of marine invertebrates, like the sponges that you see here, along the root of a mangrove. And along, in another location, in Key Largo specifically, another part of the Keys, you see a different type of habitat, in that in the mangrove forest there, these Aptasia are growing along the entire length of the root, covering the whole thing, and very little invertebrates grow alongside it. So it's really interesting to see these differences in where they grow. And I've been deploying slides and tiles, ceramic tiles, to collect biofilm over time to see whether those would be able to induce settlement and metamorphosis. The other project that I'm working on in the lab currently is to develop transcriptomic resources for Aptasia larvae. The lab here, my lab mate has already completed the adult transcriptome, and that has been published already and available to the community. And we thought it'd be interesting and useful to develop a larval transcriptome as well, because the larvae may express genes that are not expressed in the adults. Specifically, what if we are able to find GLW-my neuropeptide precursors that may mediate settlement and metamorphosis? And that way we would be able to sort of hit two birds with one stone here, figure out the settlement and metamorphosis question, as well as find some new genes in this larval transcriptome. And the larval gene expression may also differ from that of adults during Symbiodinium uptake, as well as under thermal stress. And so in our lab, we actually we do a lot of experiments with thermal stress as well, exposing anemones to heat stress, light stress as well, to determine how their health and their symbiosis might be affected. And so the goals here are to establish a baseline larval transcriptome to begin with, and then conduct a global comparison of the adult and the larval transcriptome, and then utilize RNA-seq to examine gene expression differences in larvae exposed to compatible or incompatible algae, as not all symbionts are the same, and not all of them can be taken up by the larva. And this is just a DIC image of a larva with the Symbiodinium cells in it. And so that's essentially so far our current research in the lab to develop these genetic development and molecular tools for the system. And so with that, I'd like to thank all of these people at the various institutions that I've worked at or conducted field studies at and my funding resources. So thank you very much for your attention.